Yeah.
But I think a lot of you guys are also living in places that's kind of crazy right now. All right, so I am going to do my best to do this today. I'm gonna pull up here the lecture, and I think I should be able to share the PowerPoints with you. Let's try. Also, if you experience any technical difficulties during the lecture, don't worry. I'm going to record and post everything. So if you don't worry if you do. And so far, let me see. So far, so good. Nobody has emailed me yet that they're having a problem getting in. So that's a good thing. All right, let me see here if I can do this. Okay. We will go to this and we will go to that. And we will go to this share. Okay, 1 second. Let me get through. We already went over this stuff, so let's go past this. Yeah, phenology assignment. Oh, speaking of phenology assignment, a lot of people have been emailing me. We'll wait this for a second. A lot of people have been emailing me because they have concerns because they're having a hard time getting to where they need to get to do their phenology assignment. Either your area has been told to stay inside and you just feel uncomfortable going out to take photos of trees. For right now, just hold off on the phenology assignment. Don't feel stressed by going to have to take pictures of trees. If that's causing you stress, that makes me very sad. The point is though, for right now, don't worry about it. I'm going to try to think of a way to adopt the phenology assignment to accommodate us in this new situation. Luckily, a lot of you should already have had a lot of pictures that you've taken over the course of the last few weeks, so we'll figure it out together.
1 second, I'm checking.
Oh, another thing I should mention is mute. So all of you guys listening to me right now, you should have the ability to mute yourself. So this is something, if you go to the top of your computer or something, you can press this thing that says mute me. But all of you guys should mute yourself. So if you say anything horrible about me, I can't hear, okay? Yeah, so please mute yourself. My husband recently took a class on Saturday and he realized he wasn't on mute when he yawned really big. It was like a oh, and the professor was like, I hear somebody moaning. So please mute yourself, or if not, be very nice and quiet if you aren't muted. Okay, cool. Now how do I have this here. I think you guys can see. Oh, and we also have chat. So feel free to chat too. But can you see the PowerPoint slide right now? Yay! All right. Thank you, Ashley. Thank you, Candy. Yes. Thank you, Emil. Okay, great. So you guys can see it. So I did that right. Correct. Cool. All right. Well, before we get started, hi, Stevens. I will say thank you guys so much for coming this morning. And I know it's a very, very weird time we're living in right now, but we'll get through it. And it's going to be okay. Luckily, it's happening during technology era. Like at least we can continue our studies while this whole crazy time is going on. But try not to be too stressed out. I myself, I see all these posts on social media like I'm so bored. I have nothing to do, and meanwhile my work has gone nuts in this last week. I have been just processing so much lab work that I was able to take home with me here in my house, so it's like this room now is like my classroom and my lab and yeah. So it's been very, very crazy for me. I hope, however you have to adapt, your day-to-day life has been okay, and I hope you're not getting driven crazy by your family members or whoever you live with, having to be with them all the time. So just hang in there. don't get discouraged, and we'll get through this. And if we have to, we have to do it on virtual, that's fine. We'll get through it and do that. Okay. So, I think, boy, when was the last time we were even in class? So you guys have a lab exam on Friday because it was canceled before spring break. So you guys will be doing that on Friday. That'll be interesting. You have to tell me how that goes. I'm trying right now to figure out the best way to do any assignments or anything like that via this whole virtual thing. So I'm still figuring it out, and so stay tuned on that as well. I'll figure out the best way to do that. We will still have assignments. I don't know if they're going to be during class yet, or if they're going to be not during class. I haven't figured that out yet. Exams will be via Blackboard, similar to kind of how you're going to be doing it this Friday. So, but don't worry about that for now either. I've posted a updated syllabus on Blackboard, so you'll see there that everything's been pushed back in terms of assignments that we were going to have in class. What else? Yeah, so I think we did the ferns. We covered ferns in lecture. That's right. And then we had the lecture exam, and then we went over the lecture exam, and now we have to keep going forward. Oh, and we're finally getting to the seed plants. That's where we're at now. Okay, so we've done all the spore plants. Spore plants are done. Now we're moving on to the seed plants. So let's see what we got here. And I hope I'm recording this. I think I am. It says I am. We'll see. All right, how do I go forward? Okay, right. Yes, so we're going to start to talk about the seed plants today. In particular, we're going to talk about this ovule, this amazing, amazing ovule. I think it's amazing, but we'll have to see. And just to get us oriented of where we've been before, right? So we have worked our way through the plant kingdom pretty well. We covered the non-vascular plants, the mosses, the liverworts, and hornworts. That seems like so long ago now, right? Like, oh, so far, far in the past. But yes, we did. We covered those. And we also have worked our way through the spore plants that are vascular, including the ferns, the lycopods, and the horsetails. And these are the things that you guys will be covering on your exam on Friday for lab. And you have a study guide for that. Sorry, I'm just pulling up my PowerPoint slide on my I have like 10 screens in front of me. So I'm going to pull up my PowerPoint slide on my other computer as well so that I know what is coming when I'm leading you through this lecture. 1 second. 1 second. Okay. Yes, so that's stuff that'll be covered on your lab exam on Friday. But now we're getting into the seed plants, right? Yay, seed plants. So we're gonna leave. Oh, can I? Oh, watch. I can annotate. Let's see. Oh wait, no, hold on. Go back. Here, watch. Let me see. Don't I? Yeah. Okay, watch. I can annotate. Ooh. So we did this. Yeah, we did this. Yeah, and now we're going here. Whoa. Okay. Sorry. I think that's fun. Sorry. No more annotating. Okay. So, oh no, go away. Go away, annotate. Okay. Yes. So now we're going into the seed plants. The first seed plants we're going to talk about are a type of seed plant called a gymnosperm. And then we will finish the course. Believe it or not, we're almost done with this course, which is amazing because it seems like a very long semester. But yeah, we are going from the gymnosperms first, which is the conifers, cycads, and ginkgo, and then on to the angiosperms, which is the flowering plants, which is the majority of plants. So similar to the first lecture we had, seeds are super important to people. Well, plants are super important to people, but in particular, seeds. So we really, really do eat a lot of plant products, in particular seeds. So this image here is just showing you kind of a grouping of common seeds that you might eat. So seeds are very, very important things for all animals, not just humans, but all animals, because they provide a nice food source for all animals. And as we talked about before, seed plants are the main plants that are in the plant kingdom, in particular the angiosperms. So the angiosperms are the flowering plants, but there's also non-flowering plants that bear seeds, and those are the majority of the plants that are in the plant kingdom. And so if seed plants are the majority of plants in the plant kingdom, then we have to ask as biologists, like, well, so why is that? Why are so many plants seed bearing? And how can seeds be evolutionarily advantageous to plants? So part of the reason why seeds work very well is because they have a really good survival value. They provide the embryo with additional protection because the embryo is now inside of a seed. And the seed itself provides a food source for the embryo. Part of the reason why we also eat seeds, because they have a lot of food for us in there. So seeds have a much better advantage in terms of selection, like natural selection, over plants that just shed spores. So if you have a choice between they don't have a choice, but evolutionarily speaking, if you're a plant that sheds spores as versus a plant that sheds seeds, if you shed seeds, your progeny or your baby will have a much better chance to survive because you're providing it protection and food. And also, just a reminder for you guys to mute. So if you are coming into class a little late, hi, welcome, thank you for coming. Please go and figure out the way to mute. Somebody just texted or asked if I'm taking attendance. I think I am. I'm going to try to based on this recording, but I'm obviously not going to like call everyone like, hey. It'll take too long. But maybe in subsequent classes I will do that. I still haven't figured that out yet. Okay. So yes, seeds. Seeds are better at protecting their embryo and provide a food source for the embryo. So this slide should look a little familiar. It's kind of like the slide we saw when we were talking about the significance of vascular plants moving onto land, right? Or the significance of plants moving onto land and then later becoming vascular. Similarly, when plants evolved seeds, we see a big change in terms of animals. So for example, if we look here, this around, let's say around 400 million years ago, based on the fossil record, we see these seed plants, in particular gymnosperms, which we'll talk about in a second, that show up. And around that same time so if you look here, I should annotate. Let's see. So around that same time, about 400 million years ago, sorry. I really like that. It's way better than the laser thing. It's so much fun. Look, I can even do this. Oh no, it went away. Here, I was gonna do this. I was gonna say, wow, look at all these beautiful animals that also show up. They're so happy that there are seeds now to eat. Okay, sorry. I'm carried away. Yes, so to go back, so around 400 million years ago is when plants start to produce seeds. And around that time is when you start to see animal species on land becoming more complex. And part of that is because now there's more reliable food sources in the sense of seeds that the plants are producing. Okay. And if you, oh, right, see, yeah. So that's showing you, look, around 400 million years ago, you have seeds. And then if you look here, it says millions of years on the top. Right, around 400 million years ago. And that's also, look at the expansion of plants onto land. I should say, if you guys have any questions during any time, just feel free to chat box it. Or you could shout it out, but it might be easier to chat box it. Okay, let's keep going. Yes, so the point of all this is that the evolution, there it is on the bottom, right? And I will post these slides. In addition to posting the recordings, I'll also post the slides like normally on Blackboard. But the point is, if you see there at the bottom right, the evolution of the seed leads to a big expansion of animal diversity. And so that's very, very cool. Let me pull this up here. Yes, good. Okay, so far, when we look at, if we're talking about the evolution of seed plants, we can't forget the plants we already talked about before, because we wouldn't have a seed plant if we didn't have all the plants that were there before seed plants. So we've looked at all of these, and this is a nice way to kind of organize what's happening over evolutionary time. So on the left side, you have the type of plant, and on the right side, you have its spore type. So the type of spore that the sporophyte plant will shed from those plants. And obviously in the non-vascular plants, the majority, all of them have homosporous spores, right? One spore type, it gets released, and it grows into one type of, it grows into a gametophyte plant that grows outside the spore, right? That's all old news with those non-vascular plants. Then we got into the vascular plants, and we see a couple of different things happening, right? Like some of the plants have homosporous spores, for example, your lycopods, your horsetails, and the majority of your ferns, particularly the ferns that are terrestrial. And then you start to see the occurrence of this heterosporous nature, right? So Selaginella is the first time we see it, where you have microspores and megaspores, two different types of spores, each spore containing a gametophyte plant, right? Heterosporous. And then in the ferns also, for the aquatic ferns, we also see this heterosporous nature. Well, heterosporous spores kind of led to us having seeds. So I'll show you this. So when we look at all the spore shedding plants, you see homosporous that leads to heterosporous. But if you look at this sheet, I can't even say, seed shedding plants, right? Seed shedding plants by nature have heterosporous spores. And that's because this is like a major point of the course of this whole course, is that this is a reduction in the gametophyte plant, going from plants that have gametophyte plants living in nature, growing in nature, to plants that have gametophyte plants inside of spores. And then when you get to the seed plants that have gametophyte plants even more protected inside of a seed, particularly the female gametophyte plant. So the point of this slide is just to note that there's a reason why we're learning about all this homosporous versus heterosporous. And basically, seeds are like the ultimate version of a heterosporous spore. That's the way to say that. Here's another way to put it right here, where it's more organized. So all seed plants are heterosporous, which is nice because now we don't have to worry about homosporous anymore. And you have megaspores and you have microspores, just like before, just like the other heterosporous spores we've talked about before. In the megaspore will give rise to the female gametophyte plant, and the microspore gives rise to the male gametophyte plant, just like before. But as I mentioned previously just now, that the seed plants are like an extreme form of heterospory. So in that you have this new thing that's called an ovule. we're gonna go through that now, but I'm just laying it out there before we go through it, that's kind of why it's so evolutionarily important, because you go from, plants that are homosporous to plants that are heterosporous until ultimately the seed, which is the extreme form of heterospory. Okay. So the ovule is a structure. We haven't talked about it yet because all of the plants we've had so far have been spore shedding plants. But it's a structure that has inside of it the female gametophyte plant with an egg, and the gametophyte with the egg is better protected because it's surrounded by these layers, and those layers are called the nucellus and the integument. When the egg inside of the ovule gets fertilized, that ovule will go on to become a seed. So it's a seed is basically a structure that has inside of it a zygote, which will grow into an embryo. And as I said, that's the result of sperm from the male gametophyte plant fertilizing the egg from the female gametophyte plant, which is now inside of an ovule. And when that happens, the ovule will undergo chemical changes and turn into a seed. So one way to think about it, I know it's kind of weird, but like, you've seen like animals, like if an animal gets pregnant, right, it changes structure, right? Still, like, so you have a person, like an animal, let's say an animal like a cat or something that gets pregnant, its body's going to change to help accommodate the zygote or embryo growing, right? And so it's kind of the same idea. So a question popped up here. Can you say that a seed is like a fertilized ovule? That's a really good question, and it's something that always comes up in this course. I would advise not to think about it like that, but yes, technically. So if you have an ovule, inside the ovule is the female gametophyte and the egg, right? And when the egg in that female gametophyte gets fertilized, what was the ovule will turn into the seed. But an ovule is an ovule and a seed is a seed. So sometimes so something just popped up right now. It's like, so the ovule is like the uterus that holds the egg. Kind of, yeah. Except the difference is that a seed and an ovule, they're structurally very different. And so that's why I would be hesitant to call a seed like a fertilized ovule. Because, for example, you can have an ovule that has a female gametophyte in it, and the female gametophyte can get fertilized, and then that's technically an ovule with fertilized female gametophyte, right? And sometimes it won't develop into a seed. Sometimes it will just, for different reasons, not continue on. So you technically can have a fertilized ovule that is not a seed. That's kind of why I'm a little to call a seed a fertilized ovule. You have ovules that contain something, and what they contain are a female gametophyte with an egg. And then you have a seed, which contains other things, including a zygote. Yes, you can't have a seed unless you had an ovule to begin with, but I like to think of them for structural purposes: ovule with these components, seed with these components. It just makes it a little bit more straightforward. Okay. So, and let's get into this ovule stuff. So this is a really, really amazing drawing. No, I'm just kidding. It's not that great. This is a drawing of an ovule. And I think annotate might have to happen a little bit here for, let's see. And also because I like to use annotate. All right, so here's your ovule. This whole thing is the ovule, this whole thing. But notice, so inside of the ovule is the Megasporangium, right? Or another word you might see in the textbook and perhaps in your lab is nucellus. But just know the Megasporangium is the same thing. So if you see those words, don't get tripped up. It's the same thing as Megasporangium. And this is the inner part of the ovule, and this is what houses the megaspore. So you might recall from the heterosporous plants that we looked at before, right? You had microspores and megaspores, and each of those came from its own type of sporangium. Microspores came from microsporangium, megaspores came from megasporangium. So look, now the megasporangium, which we're talking about first, not the microsporangium, is inside of all these structures. It's inside of this whole structure. So that's different. Oops. How do oh, sorry. Get out of annotate. Okay. Oh my God, come on, get out of here. All right. Okay. And so inside, so here's the megasporangium, like we said. We have megasporangium. And then inside megasporangium is this functional megaspore, which in this diagram is red. And this functional megaspore is never released. It stays inside of the megasporangium. So that's also a new thing, because before, for example, in Selaginella, when you had microspores and megaspores, the spores would ultimately get released always, right? Microspores would get released from the microsporangium. Megaspores would get released from the megasporangium. But now in the seed plants, the megaspore does not get released ever. The megaspore stays inside of the megasporangium, which is part of the ovule. That's a big difference. And the megaspore is going to grow into the female gametophyte plant, and she's also going to stay inside of the megasporangium. So these are two new things for us to handle, because before you had a megaspore that would get released, and inside of the megaspore was the female gametophyte plant. Now you have a megaspore that never gets released, stays inside of that megasporangium, and grows into the female gametophyte plant while being inside. So that's different. Okay. Oh, geez. Sorry, I'm still learning how to do this here. Okay, so yeah, so we have megasporangium, then we have functional megaspore, and then these layers called integument. So integument is just a layer of protection, and it surrounds the megasporangium. So these are all the different layers of the ovule. After the egg gets fertilized, it's going to be the integument that will ultimately turn into the seed coat. So this goes back to your, ovule is a fertilized seed. Sorry, seed is a fertilized ovule. Yeah, okay, guess kind of. But for example, we call the outermost layer in the ovule the integument, and after it is on its way to becoming a seed, we call it the seed coat, right? There's a lot of chemical changes involved in transitioning that. And then there's this thing called the micropyle. The micropyle is basically a hole. It's a hole at the top that allows sperm to enter. And the sperm is going to be vectored just like before via the microspore, which we'll talk about in detail later. So parts of the ovule are megasporangium, functional megaspore, integument, and micropyle. There's 4 parts. So just to drive this new thing home, yes, the megaspore now in seed plants never leaves the megasporangium. Stays inside. This is kind of what I was talking about before. So this is to remind you of what happened in the past, right? This is from Selaginella, where we first encountered heterosporous plants. So here, the megaspore was inside of megasporangium, and ultimately was released, right? Ultimately was released. And that megaspore would go on. Do I have? Yes. That megaspore would go on being free out in the world with the female gametophyte inside. And that's what we're used to before. That's what used to happen before. But that no longer happens now in the seed plants. The seed plants have the Megasporangium containing the megaspore always. Oh yeah, and then this is another difference. So far in the seed plants, the main difference is that there's now an ovule and now the megaspore is never released. But another thing I should point out is, let me see. So it's hard to see in this image because it's it only shows 3. But remember when we talked about meiosis, how every time you have the creation of spores, you end up with a tetrad of spores, right? A tetrad or 4. Well, in the seed plants, now when you have meiosis occur, only one of the spores of the tetrad will actually be functional. So there's only one megaspore. Right. Well, yeah, don't worry so much about the top thing, but there's only one megaspore site. Sorry. Before we had sporocytes, right, that would go on through meiosis and create a tetrad of spores, right? Diploid sporocyte goes through meiosis and ends up with a tetrad of spores that are all haploid. Now we have sporocytes, particularly for the megaspore site, the female part, is going to give rise, just like before, to a tetrad, but only one of those spores is going to survive. And that's why we call it functional megaspore. So if you go back here, right, when I said the parts of the ovule, which is the Megasporangium, functional megaspore, integument, and micropyle, the reason we say functional megaspore is because there were 4 megaspores after meiosis, right? A tetrad. But 3 of those get discarded and only one will go on to becoming the actual megaspore, and that's why we call it functional megaspore. Okay. Right. So only one of those spores survives. Right. And just as a recall, right, this is what I was saying before. Sorry, I didn't realize I actually had slides for this. In Selaginella, there was obviously many megaspores per Megasporangium, 4, meaning 4, but now only 1. All right. Talking about that functional megaspore, just a reminder to please mute. So here we got to talk a little bit about this functional megaspore. So there's only one megaspore inside of the Megasporangium. And how does this happen? So there's a process that happens to create this functional megaspore. Before we talk about that process, though, the main point of this slide is that if you have one functional megaspore and it's that functional megaspore that gives rise to that female gametophyte plant, right? So you have the process of meiosis, you have a tetrad of spores, and then one of those spores will go on to become the female gametophyte. So I had a question up here about what is the evolutionary advantage of having this one functional megaspore. And as we'll see going in the next few slides, part of the reason is because we have unequal cytokinesis now with these spores. And so I think this will become more clear on the next few slides. The point is that during meiosis, the majority of the cellular products get given to one of the members of the tetrad, one of the spores. And all the other spores basically get very little of the cellular components. And that makes that what becomes the functional megaspore more robust. I think it'll become clear in the next few slides. If not, feel free to ask me again. Okay. Yes. So this is the process of going to that megaspore. And the process is Megaspore Genesis. So Genesis means the beginning of. So you can think of Megaspore Genesis as like the beginning of the megaspore. In this case, the functional megaspore. So let's walk through what this looks like in terms of what is Megaspore Genesis. So here we have our ovule, and inside of our ovule we have one sporocyte, right? There's only one sporocyte, which is 2N, that's inside of the ovule. And that's that black dot right above that box. That sporocyte is going to undergo meiosis 1 and 2, and the result is going to be 4 haploid daughter cells or 4 spores, right? A tetrad of spores. Unlike in the spore-bearing plants, this tetrad doesn't stack up on itself like billiard balls. This tetrad actually lines up in a straight line, as shown in this diagram. And as you can see, there are 4 daughter cells there, but because of unequal cytokinesis, one of them gets the majority of the cellular components, whereas the others get very little. They get the genetic component, but they don't really get much of the other stuff. And this, so what this is showing, if I can get here, yes. So this large spore will go on to become the functional megaspore, and that functional megaspore will grow into the female gametophyte plant. So this here, oops, sorry, wow, okay, very not good at that. I guess I should practice if I'm going to be doing that more. Why is there a line there? It's weird how it shows there. But yeah, this here is the female gametophyte plant. So this functional megaspore, which is this dot, grew into this female gametophyte plant that stays inside of the ovule. Okay, how do I get out of this? Yes. Okay. Oh man. This is. Oh gosh. And I even. Of course I did. So I didn't have to draw that because I already drew it for you. So yeah. So this circle, which is much better than my hand-drawn circle, is showing the that functional megaspore, which grows into the female gametophyte plant. So there you go. Better than my drawing. So that female gametophyte plant is going to have everything that we've talked about before, right? It's going to have sex organs. It's a female gametophyte plant. Gametophyte. Phyte means plant. Gamete phyte. Plant that makes gametes, right? So even though the female gametophyte plant is inside the ovule now, it's still going to have an archegonium, it's still going to have egg, it's going to have all that stuff because it's what it does. When the egg of that female gametophyte plant gets fertilized, then that's going to be what leads to your new sporophyte generation, which is the embryo, and that's going to grow inside the female gametophyte plant. And as I mentioned, the integument is this layer around the whole thing, and it's open just at the top, which is the micropyle, to let sperm inside. Ovule apex modification. I don't know what that means. I don't know what that means. But I think it's basically, the point is that you have the female gametophyte inside of the ovule. The ovule is protected. The female gametophyte plant is protected by the integument. There's a hole. Oh, it's probably this. Oh, yes. It's a hole. Yes. It's a hole at the integument. The micropyle is a hole at the apex of the integument that allows microspores and sperm to enter. That's what that ovule apex modification means. Yes. All right. We'll talk about that more on subsequent slides. Let's see how we're doing here. Oh, good. Yeah. Ooh, we're almost done. We're not that far away from being done. Hey. So here, this is a seed that we're looking at here. So if I can go back, sorry, not to make you dizzy. Right. So remember the first one, right? This is an ovule with a sporocyte inside. Sporocyte is going to undergo meiosis and cytokinesis to unequal cytokinesis to make a functional megaspore. That functional megaspore, which is that functional megaspore, right, is going to grow via mitosis to make this female gametophyte plant that is now inside of the ovule, right? That's what we've talked about so far. So if we, there we go. Okay. So once that female gametophyte plant's egg gets fertilized, the embryo is going to grow, and what was the ovule is going to go on to become the seed. So initially, particularly in the gymnosperms, which is what we're going to talk about first, starting next class. So here you can see this is a seed. This here, this little thing here, is the integument that is on its way to. Now, granted, this is a slide, right? So like somebody had to cut the seed in half. That's why it's broken like that. But this is the integument that's going to go on to become the seed coat. This thing in the middle here that says 2N is the embryo, the result of egg and sperm coming together. And this stuff surrounding the embryo that's haploid is what was the female gametophyte plant. So basically, particularly in the gymnosperms, the embryo is going to basically consume the female gametophyte plant as it grows until it is able to make a root and things like that. We'll talk a lot. We're going to talk more and more about this whole ovule and seed thing. This class is mostly just kind of getting you used to the idea of the ovule, so that when we move on to the other things, you have this like baseline of like what a seed plant is and why we call it a seed plant, and it's because of this ovule and seed thing. So remember on this slide, I didn't know what I meant by ovule apex modification. Right, I remembered now. So it's kind of like, how did we get this ovule? What happened in terms of evolutionarily, like how did we get to an ovule? And honestly, we don't really know exactly what happened. Unfortunately, like flowers and ovules, and they don't really preserve. Sometimes they don't super preserve great because they're very fleshy. So we don't have a complete fossil record in terms of like this is the steps of becoming an ovule. We're pretty sure that the oldest ovules that we have evidence for, like I said, is around 400 million years ago. And so based on our fossil record, we have some of an idea of what we think happened. And so, okay, so this is probably, based on the fossil record, this is what a primitive ovule might have looked like. So let me see if I could walk you through this here. Right, so this whole thing here with these weird, with these weird arms and things is what the fossil record shows us that like a primitive ovule would have looked like. And if you notice, these little arm-like things are the integument, but it's not fused yet, right? They don't completely, it's not covered with just a hole at the top of a micropyle. It's like these weird kind of branch-like things that come around the ovule, but they don't actually like protect it. It's just kind of like they're like around it like that. And we think, based on the fossil record, that what happened is there were these kind of like what we call cupules, because we don't really know what to call them. But basically these, so you would have had these ovules, and you would have had these sterile structures kind of protecting it a little bit, which we call cupules. But basically these like parts of the plant, like if my head was an ovule, there were like these kind of branch things that were kind of protecting it, but not really. It's not like nice protection like that. It's just kind of like, like a little bit of protection. And what we think happened is that some of the cupules kind of grew more. Remember the whole overtopping thing of like how we think that leaves were made? It's similar to that process. But the point is, don't get too bogged down about this. The point is that we think that there was ovules, and we think that there were structures around the ovule that were kind of providing okay protection, but that the plants that those structures got closer together provided the ovule with better protection. So the cupules are sterile, the ovule is obviously fertile, and this is kind of, rather than me trying to explain it, this is what we're talking about here. So this is what we think, based on the fossil record, this is what we think happened, right? Initially, you had something like this with the ovule inside and these kind of like weird little arm things kind of protecting it, but not like super protecting it. Over evolutionary time, the ovules that had the little arm things more fused did a better job at protecting the ovule. And then over time, and so this is how the integument was made, right? Over time, these structures would have fused together like this, and then ultimately you end up with something like this, where you have this layer of protection surrounding the Megasporangium, right? With a hole at the top to allow sperm to enter, but otherwise being completely protected from the elements. Okay. Boy, I did not mean to spend that much time on that. And yeah, that hole is the micropyle, which allows sperm to enter. Okay, we're almost done. We're almost done. Thank you. You're hanging in there. I know it's morning. Ugh. Okay. So yes. So this goes back to what I said at the beginning of the lecture, in that the majority of plants are seed plants, right? So in this pie chart, green indicates the seed plants, and seed plants are over 90% of all plants that we know of. And so this seed, ovule and seed, works really, really well, because if it didn't work so well, we wouldn't have that many seed plants. So by evolving an ovule and ultimately a seed, you get a huge evolutionary advantage compared to not having that. And so there are two types of seed plants. There are the Gymnosperms and the Angiosperms. And when you look at those, the majority of seed plants are flowering plants, right? So in this pie chart, red indicates the Gymnosperms, and then the kind of lavender color is the Angiosperms. And so this is all seed plants taken together. Very few of them are Gymnosperms, many of them are flowering plants. All right, so just last thing is some terms, like, so I keep saying Gymnosperm and Angiosperm, and let's talk about what those terms actually mean. So Gymnosperm, gymno is Greek, which means naked, and sperm refers to a seed. That's a Greek word for seed. And so gymnosperms are naked seeds. And what that means is if you have a naked seed, you have a naked ovule also. So if you have an ovule that's naked and it gets fertilized and becomes a seed, then you have a seed that's naked. So we say naked seed, but it's also naked ovule. And the point is that the ovules are not enclosed in anything, right? The ovules are not enclosed. Oh my God, I'm very bad at that. Can I draw a line? Oh my gosh. This is going to be fun to watch later. Okay. Let's see if I can draw a line. Is this line? Oh, geez, forget it. Oh no. I have to practice on my annotating, guys. Sorry. The point is, though, let's see if I can get to my little, where's my little, here, pen. Okay, yes. So gymnosperms means naked seed, and what that means is the ovules are not enclosed in an ovary. So you can see them with the naked eye. They're exposed to the environment. And then angiosperms, which are the flowering plants, you have the ovule that is now enclosed in an ovary, which we'll talk about a lot more later. But the angiosperms are not exposed to the environment. The ovule is not exposed to the environment. Okay, so the ovule for oh God, how do I go back to that? I'm still also working on trying to, okay, here, chat, good. All right, so I got a question. So the ovule for the gymnosperms don't have an integument either, or the integument is always a part of the entire structure? Great question, great question. So we'll see on the next slide about this. So the integument is part of the ovule, So if you have a naked ovule, it still has an integument because it is part of the ovule. So good question. And I think this next slide will actually make this a little bit more clear. Let's see. Very good question, though. Yes. So here you go. This is a comparison of the ovules of gymnosperms and angiosperms. So gymnosperms, okay. So question, so the ovary have many ovules which consist of a megasporangium. Yes, exactly. So basically, gymnosperms don't have ovaries. Gymnosperms just have ovules. Angiosperms, which are the flowering plants, have an ovary, which is an additional layer of protection. Next question. Micropyle allow microspore or just a sperm? You guys are so smart, man. Okay, so we haven't talked about the sperm yet, so hold tight on that. We will go into great detail about that in the subsequent lectures. But yes, the micropyle allows the microspore, which for right now, just consider that the sperm. But the point is, you have an egg, right? You have an egg that's inside of the ovule. And in order to fertilize that, you need sperm. And so the micropyle is an opening that allows sperm to enter via the microspore. But we will talk way more about that in subsequent lectures. We don't have to get into that today. It's there's too much stuff in one day. But good questions, guys. So to wrap up here, this is a comparison of the ovules of Gymnosperms versus Angiosperms. And the point is look, so here, this is Gymnosperm, and you have one layer of integument, and inside the female gametophyte plant, oh, sorry, well, not yet in this. In this, just it hasn't grown. This is the Megaspore site, so it hasn't grown into a megaspore yet or anything. But the point is, this is a Gymnosperm ovule. It's naked in that it's not surrounded by an ovary, and it also only has one layer of integument. Here is an Angiosperm ovule. It has better protection in that it has two layers of integument. I can't draw. But additionally, it's inside of a new structure called the ovary. Now, the next few classes focus on gymnosperms, right? Gymnosperms. So don't worry too much about this slide right now. But the point of the slide is just this. The reason I'm showing you this is because of this. So the majority of plants in the plant kingdom are seed plants, right? The majority of the plants in the plant kingdom are seed plants, or green in this pie chart. But the majority of seed plants are flowering plants, or angiosperms. And part of the reason that is the case is because angiosperms have their egg better protected than gymnosperms, and that's why the majority of plants are angiosperms, because of that layer of protection. So don't worry too much about this right now. It's just to show you the comparison of why there are so many angiosperms as versus gymnosperms. But before we talk about angiosperms, we are going to talk about the gymnosperms, and the gymnosperms are also super cool, even though they don't have flashy flowers. So next class, we're going to talk about cycads, which are one type of gymnosperm. And then next class after that, we'll talk about ginkgo. And then finally, conifers, which are also gymnosperms. So this is what we're going to be handling the next few classes. And if you haven't already, please pull up the revised syllabus. Yeah, but that's all I have today for you guys. And if you have any additional questions or you want to talk about anything in particular, I'm here. But otherwise, thank you guys so much for coming today, and sorry if I was a little clunky. This is, like I said, my first time doing that, and it's a little weird. But thank you guys so much for coming. And if you have any questions right now, I'm happy to answer them. And then if not now, then in future, send me an email in the future. So I had a question pop up here about assignments. So I'm still figuring out how to do that. Yeah, so we have, via our class, we have Blackboard, and Blackboard has some ways to do things like tests, but it's actually really clunky on my end. Like, it's not easy for me to just upload an assignment onto Blackboard and it becomes an assignment that you guys can actually interact with. It requires me to load every single question manually, and so there's also other ways I might deal with it. I might do Google Forms. WebEx also has a way to have you guys kind of interact like in a survey type thing. So I haven't figured out how to do the assignments yet, but we will have them. I just don't know what format that's going to be. Whether it's something that you guys do on your own and turn it into me separately via Blackboard, or if it's something that we can do during regular class time. I haven't figured that out yet. That's why I pushed all the assignments back for now, because I haven't figured it out yet. Any other questions? Yes. Okay. Yes. Good question. Yeah. So many of you have emailed me about the phenology assignment because it's becoming difficult for you to do it based on the current situation. So for right now, you don't need to continue taking photos. If you can continue taking photos and if you enjoy going out and taking them, that's fine. But if it's becoming very problematic for you to go out and take photos of your plants, stop taking photos for now. I'm thinking about what the best way to proceed with the phenology assignment is. I may just make it that it's a shorter phenology assignment and that, you guys have been taking pictures up until now, and maybe that will be the photos and that's it. Or maybe I'm going to think of another way to make it more something you can do digitally. I'm still thinking about that. Yeah, so there's definitely like yeah, don't be going to campus if you guys can avoid it. But yes, please pause the phenology assignment for right now. Meaning please stop taking photos. That doesn't mean that you still don't have material to turn in for the deadline, but don't continue taking photos now if it's problematic for you, please. No problem. Any other questions? Let's see. No problem. Thank you guys so much. I really appreciate you guys coming in today. And like I said, I'll be posting this awesome lecture. I don't know how I'm going to post it yet. It'll either be posted via WebEx or on Blackboard, and I'll post the slides on Blackboard as well. If you don't have any other questions, I'm going to head out, and feel free to email me any questions you guys have. And again, really appreciate you guys coming in today. So take care of yourself, guys. Don't be too stressed out, and just one foot in front of the other. Just keep going. All right. All right, guys. Take care.